So, welcome again, everybody, to our monthly transplant webinar on pancreatic cancer. I'm Federico Canzian, and today it is my pleasure and privilege to introduce our speaker, Bruno Sainz. So, Bruno is based in Madrid, Spain, and he is a group leader of the Cancer Stem Cells and Fibroinflammatory Microenvironment Group at the Biomedical Research Institute Sols-Morreale, and also head of the Biomarkers and Personalized Approach to Cancer, or BioPAC group, at the Ramón y Cajal Health Research Institute, which is quite a mouthful. And it's also, as multiple people pointed out, and I can confirm, he is a very nice person. And so Bruno's research is focusing on pancreatic cancer stem cells, animal models, with a strong focus on translational research. And today he's going to give us an overview and talk about his research on pancreatic cancer stem cells and transcriptome in relation to PDAC. So I leave you the floor and everybody enjoy the seminar.
Thank you, Federico, for the invitation. As you all know, Ingrid and I lead the webinar, so it's nice to be on the other side now and to give you guys a perspective of what we're doing in the laboratory here in Madrid. So as Federico already mentioned, we're part of two institutes, the IIBm and the IRYCIS, both where we have two laboratories that are mainly focused on translational research with pancreatic cancer stem cells being the focus. So obviously we don't need to get into all the details of what pancreatic cancer is. We've been seeing this now for the last 2 years in all the webinars, but we can say is that when we compare PDAC survival rates to other tumors, we are not advancing the same way as we would like to be. Still, we have 5-year survival rates around 12%, anywhere between 10 to 12% based on or in function with the country or the continent. And it's predicted that pancreatic cancer will become the second leading cause of cancer death in the US, behind only lung cancer by 2030. Now, what these predictions did not take into consideration when they were made is the advancements that we're making in the KRAS inhibitors. So we will have to see whether or not the KRAS inhibitors, once they're more clinically available to patients and used in the clinic for both resectable pancreatic cancer patients and also metastatic pancreatic cancer patients, whether or not these statistics and 5-year survival rates will change. So this is still in the work, and we will see how this evolves in the future. But nonetheless, we can say that pancreatic cancer is one of the most deleterious and most lethal solid tumors that we know. And I think if we're going to make an impact on pancreatic cancer and our understanding of pancreatic cancer, we really need to take a translational approach, a research approach for battling this disease, anywhere from preclinical models to animal studies. Artificial intelligence is really making a splash, also moving into human studies, translation to patients, and obviously translation to the population, which is going to be probably the biggest effect. So I think all of us that participate in transplant and this cost action fall into one of these categories. And I think together, this is one of the beauties of these cost actions, that we can come together, share our expertise, and try to fill in the gaps that we may have, such that we make the full circle of this continuum to make sure that we have an effect or translation to the patients. So in my laboratory, as Federico mentioned, we're the Cancer Stem Cell and Fibroinflammatory Microenvironment Group at the IIBm, and also the BioPAC group at IRYCIS, and we tackle pancreatic cancer from a number of angles. And we try to understand the tumor biology and the tumor microenvironment of this tumor. It gives us an idea of the heterogeneity that exists within the tumor. Omics, we'll talk a lot about this today, our omics approaches to try to understand, very basic omics approaches, when we compare that to other people in the cost action that do much more sophisticated omics approaches. And how we've been able to understand the tumor and the pancreatic cells at the omic level to try to develop new drug therapies, understand their metabolism, with the ultimate goal is to try to take this to the patient and what we know as precision medicine or personalized regimens. So the tumor makeup or intratumoral heterogeneity, without a doubt, we understand that pancreatic cancer is extremely heterogeneous. In fact, the pancreatic tumor cells represent a very small population or percentage of the tumor, while the rest of the tumor is primarily made up of the tumor microenvironment. So we know that tumors are extremely heterogeneous, which really makes a one-size-fits-all approach for tackling this tumor quite impossible. And so we can't treat each tumor the exact same way because each tumor is extremely different, not only at the level of percentage of tumor cells and stroma, but also at the composition of the tumor cells. And we believe that the cancer stem cell concept or model nicely explains tumor heterogeneity. For those of you who are not familiar with cancer stem cells, they represent a very small, rare population of cells within the tumor. They're referred to often as cancer stem cells, also oftentimes also referred to as the cell of origin or the tumor initiating cell. They sit at the apex of what we call the tumor hierarchy. These cancer stem cells are similar to normal stem cells in some ways, for example, that they have unlimited self-renewal, so they can undergo both symmetric and asymmetric division, symmetric division always giving rise and maintaining the cancer stem cell pool, and asymmetric division giving rise to all the other progeny cells that we find within the tumor. These are the cells that really make up the bulk of the tumor, while the cancer stem cells represent a very small percentage of these cells. But the cancer stem cells are the ones that are most pluripotent, are the ones that are extremely chemo-resistant, and the cells down at the bottom are more differentiated, and we have a greater heterogeneity within the bulk tumor cells. So cancer stem cells were first described about 32 years ago, and we've learned a lot about them since their initial discovery. And we know that they have been identified in almost all solid tumors, including pancreatic cancer. However, many questions still remain about what is the role, for example, where do cancer stem cells come from? What is their exact role in the tumor? And from my laboratory and the BioPac laboratory perspective, we ask the question, if we eliminate them, can we cure cancer? I think this is a question we've been asking ourselves for the last 20 years, and as we learn more about cancer stem cells, we know that just targeting cancer stem cells will probably not eliminate the tumor. What we have to do is target cancer stem cells, the tumor microenvironment, and also other cells within the tumor that have the ability to turn into a cancer stem cell if we attack the cancer stem cell population. So these what we call plastic or more dynamic cells within the tumor. So the way we study cancer stem cells in the laboratory has primarily been using cancer stem cell markers, and mainly proteins that are on the cell surface of these cells that allow us by different techniques, primarily flow cytometry, using antibodies to identify these cells and then pull them out of the population to study them in depth by omics analyses, also to culture them, etc., or to inject them even in animals to see their tumorigenic potential. And the way we do this, as I mentioned, is to use antibodies which target different proteins on the cell surface of cancer stem cells or pancreatic cancer stem cells. These are the most common cell surface proteins that have been used, for example, EpCAM, CD133, CXCR4, ABCD2. We also have some internal enzymes, for example, that also can identify cancer stem cells, for example, ALDH1. In the laboratory, over the last 10 years, we've identified many more cancer stem cell markers, for example, CD47, and we've identified also P2X7R and FPR2, which are on the cell surface of cancer stem cells, anthrax toxin receptor 1. And we're always looking for new cancer stem cell markers to allow us to identify them. There's a lot of heterogeneity also within the cancer stem cell population, so one marker does not identify all cancer stem cells. And so we are always looking for new markers. In 2024, we published a new marker that is expressed on the cell surface of cancer stem cells, PGLERP. It's expressed on the surface and is also secreted, and also has functions when this protein is secreted from the cancer stem cells. And this is all due to omics approaches. So since we have all of these markers that allow us to separate cancer stem cells from non-cancer stem cells, what we're able to do is see how they were transcriptionally different. And so here's just a very simple volcano plot showing genes that are upregulated or rich genes in cancer stem cell population versus the non-cancer stem cell population. And of course, as we expected, we found a number of genes that had already been described as upregulated in cancer stem cells, for example, LGR5, DCLK1, etc. But we also found this protein, PGLERP, which stands for peptidoglycan recognition protein 1, which is a protein that had not been previously described in cancer stem cells of any type of tumor, and it was primarily known to have functions in antibacterial functions. So why was it overexpressed in cancer stem cells? What role did it play? So we did a number of experiments. This project was led by Juan Carlos Lopez Hill, who was a PhD student in my laboratory, who's now a postdoc in Mariano Barbacit's laboratory at the CNIO. And what he did a number of experiments, but I'm just going to show you one key experiment here, which really shows the role of PGLERP on the surface of cancer stem cells. So we have wild type cells that are derived from the KPC model. So these are murine pancreatic cancer cells that when you inject them into immunocompetent mice, they form tumors. We overexpressed PGLERP to see what would happen if it was overexpressed. And then we edited by CRISPR-Cas9 editing PGLERP to see what would happen if the cancer stem cells no longer express PGLERP on their cell surface. And what we saw with the wild type and the overexpression at different dilutions, you can see here the dilutions on the right is that the tumors obviously formed at all the dilutions that were tested, differences in size based on the number of cells that we injected. But when we injected cells that did not express PGLERP, we saw that at none of the dilutions tested were the cells able to form tumors in immunocompetent mice. So we repeated this experiment in immunodeficient mice. So we took the very last limiting dilution, which was 1,000 cells, and then we injected these immunocompromised mice, And then here you can see now that the cells that do not express PGLERP are able to form tumors. Really no differences between the groups. So this really told us that PGLERP is necessary for cancer stem cell immune evasion. When the immune system is competent and the cells do not express PGLERP, the immune system is able to attack these cells, eliminate them. But when you implant these cells in immunocompromised mice that do not have an intact immune system, especially an adaptive immune system, then these cells are able to proliferate and form tumors. So this is really a nice example that cancer stem cells, the proteins that they express on their cell surface do have functions, and in this case, PGLERP has a function or gives the cancer stem cells a capacity for immune evasion. We've gone further with this to really try to understand the role of PGLERP in immune evasion, and also recently new data showing that PGLERP also plays a role in subtype switching. So we know that in pancreatic cancer, there are two main subtypes that exist: the progenitor classical subtype and the basal quasi-mesenchymal squamous subtype that we find present in pancreatic cancer. And what Juan Carlos was able to show is that when you have PGLERP at normal levels, you have high expression of EpCAM, low expression of Imentin, more of a classical epithelial or progenitor-like tumor environment or tumor cells. But when you overexpress PGLERP, so here we induced expression of PGLERP with oxycycline, you can see massive upregulation in PGLERP. We also see, in parallel with this, a decrease in EpCAM and an increase in Vimentin. And of course, if you look at GATA6, which is the main marker that is used by a number of laboratories to show the difference between a classical or epithelial-like tumor versus a more basal-like tumor, you can see that when we induce PGLERP, GATA6 levels decrease by immunohistochemical staining and also by qPCR, we see a very nice downregulation of PGLERP. Of course, if you look at the cells, this is a key indicator here. You can see how the cells are more mesenchymal, more non-epithelial state here compared to the wild type cells. If we perform GSEA analysis on RNA-seq that were performed in these cells, and we see upregulation in the PGLERP overexpression cells of genes or pathways involved in motility, migration, EMT, etc. And of course, if you look at some other pathways, for example, the Moffitt classical, you can see that this is enriched in the wild type cells, but not in the overexpression cells. And when we overexpress, we see an enrichment in pathways, for example, the basal, or here I'm showing the EMT, the Yosef EMT T1 invasive pathway. So indeed, PGLERP on the cell surface of cancer stem cells provides an immune escape mechanism, but also when PGLERP is secreted into the tumor microenvironment, it can promote this subtype switching from a classical to a more basal-like. So it's something we're still continuing to study to understand exactly when this happens during tumor evolution and whether or not we can use this to our advantage. So all of these cell surface receptors have been quite useful, but they do have their caveats. You have to use antibodies to identify them. They require flow cytometry to be able to sort them, etc. So for many, many years, we were asking whether or not we could find a more simplistic or more simple biomarker for cancer stem cells, and maybe a biomarker that was not specific for pancreatic cancer, but could also be universal or applied to other types of cancers. So in 2014, quite some time ago, almost 10 years ago, we published the identification of another biomarker within cancer stem cells known as autofluorescence. Now we'll go into the details of this. This is now a very old, I don't want to say old, but a publication that's already been on the market for quite some time. But what we saw, especially at Miranda at the CNIO, is that cancer stem cells also have inherent or intracellular vesicles within their cytoplasm that actually autofluoresce on their own. And this autofluorescence can be detected by flow cytometry. You can see here the tumor. Would be the autofluorescence peak. This is our way of identifying the autofluorescent positive cells. And not only is this great for actually tracking in real time and also be able to sort pancreatic cancer stem cells without the need for antibodies. But the question also we had is whether or not, what was this autofluorescence, and whether or not this accumulation within these autofluorescent vesicles had a specific or important biological consequence for the pancreatic cancer stem cells. So later we identified that these vesicles were coated in ABCG2. You can see here ABCG2 fused to M-cherry nicely coat the autofluorescent vesicles, and subsequently it was identified that what they were concentrating in these autofluorescent vesicles was nothing more than riboflavin or vitamin B2. And vitamin B2 or riboflavin at high enough concentrations is actually autofluorescent when excited with a blue laser. And you can see here in this time-lapse video is that the autofluorescent vesicles, so these are primary cultures of pancreatic cancer cells. The nucleus has been stained with M-cherry, and you can see here the formation of the appearance of the autofluorescent vesicle. It's a very dynamic process. They appear and they disappear, probably indicating the different states of the cancer stem cells within a period of time in both in culture and in vivo. And for many years, we wanted to see whether or not, specific between 2015 and 2019, whether or not this autofluorescent phenotype was specific just to pancreatic cancer. So we analyzed 145 different tumors in collaboration with local hospitals here in Madrid to see whether or not we could detect autofluorescent cancer stem cells in other types of tumors. And again, we find a large majority of the PDAC tumors were autofluorescent positive, also colorectal cancer, including also pancreatic neuroendocrine tumors. This was carried out by Sols-Morreale. And you can see here that we have a wide range of autofluorescent positive cells, or percentage of autofluorescent positive cells that can be detected in different types of tumors. For example, here in rectum, some patients that have very high levels of autofluorescent cancer stem cells in their tumors, and also in, for example, colorectal cancer, where we had 75 patients that we were able to analyze, but we have some patients that have very, very high levels or very high percentages of autofluorescent positive cells within their tumors. And since we had 75 patients, and forgive me for moving just briefly into colorectal cancer, I'll jump right back to pancreatic cancer, which is our main focus. But since we had 75 patients, we asked whether or not this autofluorescence could give us any idea about prognosis, recurrence-free survival, etc., in this cohort of patients. So we were able to divide the patients into patients that had low levels of autofluorescence and high levels of autofluorescence. And in the end, what we were able to see is that it's actually a very good predictive marker when combined with positive lymph nodes for colorectal cancer to predict the probability of recurrence-free survival 5 years after surgical resection. So we know that colorectal cancer, the majority of patients can undergo surgical resection, but 25% of those patients will relapse or have tumor recurrence 5 years after surgical resection. And to date, the best biomarker or the best predictor for whether or not those patients will have recurrence 5 years after surgical resection is whether or not they have positive lymph nodes at the time of surgery. But if we combine both the presence of high autofluorescence population and positive lymph nodes, we have a much better ability to predict which patients are the ones that are going to relapse in 5 years. So these are really the patients that we have to pay attention to. These are the patients that might have to undergo a longer cycle of post-operative chemotherapy. These are the patients that are of high interest. And now combining these two markers, we can better identify those patients. So omics has been a huge part, obviously, of the last 20 years in different scenarios. Now we're in the era of single-cell RNA-seq, spatial transcriptomics, etc. But again, we've done a lot of very basic omics approaches trying to understand cancer stem cells, comparing them to non-cancer stem cells and also embryonic stem cells. And we've published over the course of the last 10 years that, for example, pancreatic cancer stem cells, their DNA is hypermethylated compared to non-cancer stem cells, which is quite interesting. It regulates a lot of microRNA clusters within the genome. At the microRNA profile, we've also shown that these microRNA clusters are very different between cancer stem cells and non-cancer stem cells. And we've also obviously performed traditional RNA-seq analyses. All of these approaches really just allow you to generate a list of putative targets that better defined or are characteristic or associated with the cancer stem cell population versus the non-cancer stem cell population. So we've done this for a number of patient samples, separating cancer stem cells from non-cancer stem cells by traditional monolayer or sphere cultures or using our different markers. And combining all of these data over the course of the last 5-6 years, we've been able to come up with a cancer stem cell signature and a non-cancer stem cell signature. Basically, we've identified 112 genes that are overexpressed in cancer stem cells and another 101 genes that are underexpressed in cancer stem cells. Or the other way of looking at it is 101 genes that are overexpressed in non-cancer stem cells. And this allows us to try to stratify patients. We can apply these signatures to the publicly available databases. So here are 5 publicly available databases where we've applied the cancer stem cell signature and the non-cancer stem cell signature. And what really pops out is the difference at the metabolic level of these tumors. So patients that we can classify as a enriched cancer stem cell tumor, what we see is activation of pathways involved in oxidative phosphorylation, fatty acid metabolism. Basically, these patients really have a metabolically active tumors at the level of their mitochondria. They're using their mitochondria for the acquisition for their energy requirements of production of ATP. While the non-cancer stem cell signature identifies patients that have a different metabolic component or profile. These patients are more dependent on glycolysis probably for their energy requirements. So we've taken this gene set, these 213 genes, and in collaboration with Julio, who runs the BioPac laboratory at the IRYCIS, and Ignacio Ruz, who's our stellar pathologist at the hospital. We've taken these 213 genes, we've done a signature reduction on them and reduced down to just 23 genes that allow us to also at the omics level efficiently classify these patients the same way as we were achieving with the 213 genes. And with just now a 23 gene signature, what we can do is very simple cybergreen qPCR-based assays. We can, from RNA extracted from either formalin-fixed paraffin-embedded sections or from fresh pancreatic tissue, we can perform a cybergreen qPCR within 1 day, and then working with an algorithm that we've designed, classify these patients. And we can do this with just one patient as a cancer stem cell enriched tumor or non-cancer stem cell enriched tumor, which we call other. And so, for example, this is a very small cohort of FFPE samples, 29 patients, where we've done this, classified them as cancer stem cell enriched or other, and we can see that the cancer stem cell enriched tumors have worse overall survival over time, and very small cohort, but in fact, being a very small cohort, the fact that we can see these differences is quite astonishing. And applying the signature also to publicly available databases where we have response to different chemotherapies, what we've seen is the cancer stem cell tumors respond or are predicted to respond better to FOLFIRINOX or gemcitabine, while the other tumors that are not cancer stem cell enriched would only respond to gemcitabine and Abraxane. So for a lot of the tumors we have in our overall survival analysis, we have established PDXs. So we've gone back to those PDXs and we've taken a cancer stem cell PDX or an other PDX, and we see whether or not this prediction holds true. And you can see here, this is a PDX from a patient that we classified as cancer stem cell enriched, and we can see that this tumor responds both to FOLFIRINOX and Abraxane. But on the opposite side, if we classify the patient as other, you can see that their PDXs do not respond to FOLFIRINOX and only respond to gemcitabine and Abraxane. So for this patient, for example, had this patient been given FOLFIRINOX, in theory would not have had any effect And by doing this classification, we would have been able to tell the oncologist that gemcitabine and Abraxane was probably the way to go with this patient, and not FOLFIRINOX, which, as we know, also has associated with it a lot of toxicity. So our MS approaches are giving rise, hopefully, to potentially new drug therapies. I'm really focusing right now, the focus of the laboratory is on metabolism. So we've been working on metabolism for quite some time, in particular in collaboration with Patricia Sancho in Aragon and Spain. And if you look back at what I mentioned earlier, the patients that we classify as cancer stem cell enriched, we see activation of pathways involved with mitochondrial respiration, OXPHOS, and fatty acid metabolism. And this parallels very nice a study that Patricia published also about 8-9 years ago, showing that cancer stem cells indeed upregulate genes that are involved in the TCA cycle and mitochondrial OXPHOS components, etc. While the non-cancer stem cells upregulate genes involved in glycolysis. So Patricia concluded that pancreatic cancer stem cells use OXPHOS, mitochondrial respiration, to meet their energy requirements. And we have built upon this hypothesis for a number of years and a number of publications, and very compellingly show that cancer stem cells are very different than non-cancer stem cells. You can see this here. I think a picture's worth 1,000 words. These tumor electron micrographs, transition electron micrographs, TEMs, and you can see here that the cancer stem cells have very functional mitochondria, very nice cristae that you see in the mitochondria, showing or indicative of functional mitochondria that would be able to use OXPHOS, while the mitochondria that we see in non-cancer stem cells are very swollen, not well-defined cristae, and in fact we see a lot of mitochondria also present in autophagosomes, indicating mitophagy and the elimination of these dysfunctional mitochondria. So we asked if cancer stem cells are dependent on OXPHOS for their energy requirements. Could this represent an Achilles heel of the cancer stem cell population? So we teamed up with José Luis Mascareñas in Santiago de Compostela in the north of Spain, who's been one of the world leaders in organic chemistry, and he's been working on a number of years on metallo compounds. Probably the most famous metallopharma compound that we know of is cisplatin, but he's been working on a number, trying to develop a number of metallo compounds that are based on ruthenium. So you can see here this compound, IGN-111, it's a ruthenium compound. It has a bipyrimidines and tripyrimidines, and it only has one reactive site here at this position. So this reactive site is usually occupied by chlorine, and when it's in the presence of water, for example, this chlorine is switched out for water and is able to covalently bind to DNA. So José Luis had the impression that this could affect mitochondrial respiration, and we proved that in the laboratory. As Sonia took this compound, treated cancer stem cells and non-cancer stem cells with IGN-111, and what we saw is a very nice downregulation of all the genes by RNA-seq analysis that are involved in OXPHOS. So clearly indicating that IGN-111 has a very profound and very potent effect on mitochondrial respiration in the cancer stem cell population. So what we discovered is that IGN-111 actually binds to the D loop within the mitochondrial DNA. So because of its positive charge, is able to enter into the mitochondrial DNA. It binds to this D loop because of its ability to bind to free guanines that are accessible primarily in very structured DNA, which are typically found in G quadruplexes. The D loop of the mitochondrial DNA is filled with these G quadruplexes and have a lot of accessible guanines. So IGN-111 can bind to this, and by binding to this, it essentially inhibits the transcription of the mitochondrial DNA. So we don't give rise to the messengers that give rise to the 13 proteins encoded by the mitochondrial DNA that are necessary for the electron transport chain and OXPHOS. And you can see here, these are just 3 genes. The MTs are 3 genes encoded by the mitochondrial DNA. This is another gene encoded by the nucleus that is also involved in the electron transport chain, but is encoded by the nucleus. And you can see that in 2 different PDX models. These are cell lines derived from 2 different PDXs. At different doses, you can see a very nice dose-dependent reduction in the transcription of these genes from the mitochondrial DNA. Why we don't see this in the genes that also participate, that encode for proteins that also participate in OXPHOS, electron transport chain, but are encoded by the nucleus. And whether or not this is the data indicated that it was specific for cancer stem cells. And what we did is just basically marked the compound with a fluorescence, and we're able to see in which population did the compound accumulate. And we used two different markers and a combination of these two markers, CD133, and we can see here in the two different PDX models of preferential accumulation in CD133 positive population, the autofluorescence population, and when we combine these two markers, a very nice accumulation in both the CD133 autofluorescent positive population. So indicating that they are reaching the cancer stem cells. And obviously we wanted to see how this had, whether or not this had effect on tumor growth in vivo. So Laura Ruiz from the Growth Laboratory was able to set up a number of different PDX models. These are primary patient tumors that are implanted in immunocompromised mice. We take these tumors, once they grow, we take them out, we implant them into many more mice and create cohorts where we can treat them with different treatments. For example, here we're just showing the data expressed as full change in tumor growth over time in the control group, which is blue, and the group treated with IGN-111. And we can see in these 3 PDX models that it's a very cytostatic drug, especially in PDX3, where we inhibit overall the growth of these tumors over the course of 30 days of treatment. We also see that the compound is actually getting to the tumor. This is just simple ICP analysis where we are able to detect the accumulation of the tumors. You can see that once we treat the mice, we take out the tumors, we see that the compound is getting to the tumor. And once it gets to the tumor, is it doing what we predicted it to do that we were able to show in vitro? Is it inhibiting the transcription of the mitochondrial DNA genes, the messengers encoded by the mitochondrial DNA. We can see here MT, ATP6, CYTB, and D4. In all cases, we see a reduction in the transcription of these genes, while we don't see this with COX-5. So IGN-111 reaches and accumulates in the tumor and specifically inhibits the mitochondrial DNA transcription. This was great. We also wanted to see whether or not it was indeed targeting and perhaps eliminating the cancer stem cell population, which was our main target, and how we believed that the compound was working, inhibiting OXPHOS in these cells that require or that meet the energy requirements via mitochondrial respiration. And you can see here, there's a combination of markers, EpCAM to identify the human epithelial cells present within the tumor. Then we add to that population CD133 to identify the CD133 positive population, and CXCR4, which is a marker when combined with CD133, really identifies a very highly metastatic cancer stem cell population within the tumor. And in all cases, what you can see is that the tumors treated with IGN-111, we were able to eliminate the EpCAM positive, EpCAM CD133 positive, and the EpCAM CD133 and CXCR4 positive cells within these tumors. So this is sort of a snapshot of what we think IGN-111 is doing, how it's working, and where we're going with this. This is the typical cancer stem cell model. We know that cancer stem cells represent a very rare population within the tumor. These tumor cells or these cancer stem cells are responsible for chemoresistance. They have unlimited growth. We also believe that they are the cells responsible for metastasis. They have unlimited self-renewal, so they're always able to replenish the cancer stem cell pool and make sure that they're always present. And in fact, after chemoresistance, they are the cells that are responsible for relapse. Again, after chemoresistance, they survive. They can then give rise again to unlimited tumor cell growth or relapse. And also, following chemoresistance, sometimes when even we believe that the tumor has been eliminated, these cells can linger, still be around, and give rise to metastasis. This is both relapse and metastasis. And what we discovered over the years is that these cells obviously are dependent on OXPHOS to meet their energy requirements, especially when we compare them to non-cancer stem cells that have very limited cell division differentiation potential, and according to the cancer stem cell concept or theory, have no tumor formation capacity. And these cells depend on glycolysis for their energy requirements. So since cancer stem cells have very functional mitochondria, IGN-111 represents a very novel approach to target the cancer stem cells by targeting the one organelle that they have different than the non-cancer stem cells. Here they have a functional mitochondria. Within functional mitochondria, we find the mitochondrial DNA. And again, as I mentioned earlier, IGN-111 binds the D loop, inhibits the transcription of the 13 protein encoding genes from the mitochondrial DNA. Now we don't have these 13 proteins that are necessary for like the electron transporter and OXPHOS. So we have a reduction in OXPHOS, which the cancer stem cells need. So therefore we have a reduction in stemness. And if we have a reduction in stemness and less cancer stem cells, which are few tumorigenicity, then we also have a drop in tumorigenicity. And this is just shown here. The blue line is the control. The red line is the IGN-111 compound. Here is gemcitabine in green. In fact, when we combine the compound with a traditional standard of care, we also see a much more potent reduction in tumor growth. So we believe that this compound could be potentially combined with standard of care in the future. And that's exactly what we're trying to do now. We were fortunately awarded an EE transition grant from the European Innovation Council, which the goal of this grant is to take IGN-111 potentially to the clinic, at least to the steps that need to be conducted prior to the first phase 1 clinical trial, which would be toxicity, ADME-tox studies in 2 animal species, and also a GMP scaling up and manufacturing of the compound. So we hope in 2 years time from now that we will be able to have some newer data and newer advancements with this compound at the level of toxicity and also as scaling up, which would be the last steps that we need before we enter into the first phase 1 clinical trial. So with this, I will try to wrap up. We in the laboratory are trying to understand the pathology or the makeup of the tumor. So we know that the cancer stem cells play a very important role in maintaining this heterogeneity that we find within the tumor, and we believe that they also play an important role in the subtypes that we find within the tumor. So we have some tumors that are very enriched in cancer stem cells or enriched in cancer stem cells that express P2LRP, and we believe that can have an effect on the subtype that is present initially, and then once P2LRP is secreted, that can create subtype switching. This omics, as you've seen, we can apply and now stratify patients that have tumors that are enriched in cancer stem cells or not enriched in cancer stem cells. And hopefully we believe in the future that we can take this very simple 23 gene qPCR to the clinic, stratify the patient once the tumor is resected or on a biopsy, and then help guide the oncologist as to what type of treatment that patient should receive based on the classification that we're able to make. And also these classifications allow us to look at differences between different patient populations, and that has been able to open up a new line of research in the laboratory at the level of metabolism, for which we're developing new drug therapies, in particular IGN-111, to try to better combat or attack the metabolically active cancer stem cells. And again, all of these approaches and everything we do in the laboratory has the focus of eventually precision medicine and personalized regimen for patients based on proper stratification using omics approaches. And then that information we can use then to stratify patients into groups that should receive different treatment approaches. So with that, I would like to end. Of course, I want to acknowledge everyone in the group, both past and present members they are really the ones that do all the work. I'm just here trying to sell their brilliant research that they are able to generate every day in the group. Of course, I will also highlight the BioPac laboratory, which is led by Julie, also with collaboration with Alfredo, and at the EDCs we also collaborate, as I mentioned, with Ignacio Ruz, who's been helping us tremendously with the stratification of the patients. All of our other collaborators for which research I presented within the seminar are here. The group from Hospital Universitario Infanta Sofia, who led all the stratification and the detection of the autofluorescent cancer stem cells in the different tumor entities. Patricia in Aragon, José Luis Mascareñas for the IGN-111 compound, and of course also our other collaborators at the CNIO, Susana Garcia Silva and Carmen Guerra. And of course our funding sources, because without them we would not be able to do any of the science that we do in the laboratory. And of course, I wanted to thank you for your attention. I'll be more than happy to field any questions you might have on the work we're doing in the laboratory. Thank you.